Hello, my name is Dr. Teresa Duda. I'm a colleague of Dr. Sharma. I feel privileged to introduce Dr. Rameshwar Sharma, the retired Distinguished Professor of Biochemistry and Molecular Biology. Together with his group and collaborators, Dr. Sharma authored 303 publications, including 3 books, 20 monographs, and held 2 US patents. He has organized and been a plenary speaker of 22 international global symposia, amounting to the awards of almost $7.3 million. His work has been funded by the numerous awarding agencies. First, National Institute of Health, Pennsylvania Lions Club Eye Research Foundation, National Science Foundation, Damon Runyon Walter Winchell Cancer Fund, and Veterans Administration Research. He is the discoverer of the multi-limbed membrane guanylate cyclase molecule that has brought a transformational change in our understanding of the field of cellular signal transduction. Amazingly, the single molecule, via its multiple-limbed molecular structure, controls all vital sustaining processes, locked with central, sympathetic, parasympathetic nervous systems. Without it, our universe would be totally dark and lifeless. Its lifelong work was highlighted in the most recent reviews published in the Frontiers in Molecular Neuroscience journals in 2022 and 2023. So now let us talk to Dr. Sharma and learn more about his lifelong work and his successes and his troubles. Thank you. Dr. Sharma, could you please tell us about all the life-changing events that shaped your life and made you to enter the field of physiological sciences?
In 1945, accompanying my father in the Dogra regiment up in the Himalayan mountains, in Bunji Gilgit of the Jammu and Kashmir state, India, situated at an altitude of 8,202 feet above sea level, I crystallized sodium chloride. With the conscious passion set on fire, I started dreaming about becoming a scientist. On October 5, 1947, at the age of 11, I became fatherless due to the Pakistan Pathan tribal army's siege of Poonch, Jammu Tawi of the Dogra battalion when my father served as a medical doctor. Now, I had to carve my own way. The year 1953, April 25 to May 12, at the age of 17, I spent approximately 2 weeks' time with Honorable Atal Bihari Vajpayee, future Prime Minister of India. It happened during my summer vacation in the house of Daya Kishan Kotwal in Bhadrawa, Jammu. Then Mr. Kotwal was the president of Praja Parishad, a political party aligned with Jansangh that later became Bharatiya Janata Party. Honorable Vajpayee was a political fugitive hiding in Mr. Kotwal's house. Then press secretary to Dr. Shyama Prasad Mukherjee, Mr. Vajpayee on May 8, 1953 at 6:30 a.m. in a jeep to Jammu had escaped the police arrest from Mr. Sheikh Abdullah's government. Mr. Mukherjee's team was protesting the existence of the Article 370 applicable to the state of Jammu and Kashmir. The article made the state semi-autonomous, yet prohibiting the residence of any out-of-state Indian subjects. Years 1966 to 2023, my research team founded the multi-limbed membrane guanylate cyclase signal transduction pathway at the molecular, genomic, and physiological levels. The discovery underwent aggressive scientific resistance, including certain Nobel Prize knitted network members. They not only denied, even ridiculed its existence. Suddenly, in complete turnaround, 3 of these members received the Nobel Prize through the discovery of the multi-limbed membrane guanylate cyclase's second messenger, cyclic GMP, that it generates as a hormonal messenger. In the fake news controversy, the discovery raised the fierce political wrath in the USA. Geared by the prominent self-serving politician, Democrat Congressman John Dingell, it was linked with the scientific fraud. Yet, in a historic court verdict, Rameshwar Sharma versus the United States government, I won and was exonerated of any wrongdoing. The multi-limbed membrane guanylate cyclase pathway survived without any blemish, and it made a transforming scientific contribution. The pathway controlled all of the life-sustaining processes. 6. Notably, the media, including Washington Post, New York Times, and Cleveland Plain Dealer, called me, acknowledging the political and scientific wrong inflicted on me, and Mr. Malcolm Gladwell from New York Post came to interview me in my laboratory. Earlier, Mr. Gladwell had released a critical podcast on this matter. Next, even to date, the multi-limbed membrane guanylate cyclase discovery has not received any major meritorious scientific acclamation. It remains largely ignored and vaguely mentioned. In politics, interlaced with the discovery, impeded its scientific progress and caused a permanent financial and personal damage to me and my family members, my wife Joan, daughter Sanya, and son Hari. All of us never recovered.
These are really fascinating events, and every one of them deserves to be narrated alone. But can you tell us which one would you like to start narrating first?
It is the discovery of the multi-limbed membrane guanylate cyclase cellular signal transduction pathway. It took 54 years, 1969 to 2023, to accomplish this task. The discovery is monumental in the field of cell biology. Not only it established the membrane guanylate cyclase signal transduction field, it also revolutionized the arts of developing clinical therapies to the diseases linked with the physiology of endocrinology, cardiovasculature, sensory neurons, and cancer. Most significantly, sole molecule, the membrane guanylate cyclase does so via its multi-limbed structure. The interconnected limbs throughout the central sympathetic and parasympathetic systems define their physiology. To do so, they generate their common hormonal second messenger cyclic GMP. The foundational principles of this pathway were laid down by the discovery of its limb, the adrenocorticotropic hormone. Significantly, it challenged the both existing dogmas at the time. First, there was the question of the independent existence of this membrane guanylate cyclase pathway that was different from its heme-dependent soluble form. Second, the only hormonal cellular signal pathway was that of cyclic AMP. Structurally, both of these pathways are 3-component transduction systems modulated by GTP binding proteins. Thus, the key question was, did a 1-component transduction system, cyclic GMP modulate, indeed existed for the polypeptide hormone ACTH?
How did you prove that in addition to a 3-component signaling system, there existed also a 1-component transduction system?
A unique structural feature of the multi-limbed membrane guanylate cyclase transduction system is that it is encoded by multiple 7 genes, respectively termed as ANF-RGC, CNP-RGC, ROS-GC1, ROS-GC2, ON-GC, STAR-GC, and GCG, bearing 9 limbs. Each limb is coded to perform a uniquely designated physiological function. Limb 1 controls ACTH modulated steroidogenesis. Limbs 2, 3, and 4 control cardiovasculature and central nervous system plasticity. Limbs 5 and 6 vision. Limb 7 taste. Limb 8 smell and central nervous system plasticity. Limb 9 regulates electrolyte and water balance in the intestine. Thereby, without the presence of the multi-limbed membrane guanylate cyclase signaling pathway, all our life-sustaining universal world would vanish. Molecularly, the membrane guanylate cyclase is a single transmembrane spanning protein. Its extracellular domain is located outside the cell and senses hormones and other extracellular chemical signals. These signals are transmitted by the transmembrane domain to the catalytic regulatory domains to control the synthesis of cyclic-GMP in the OR catalytic domain. Together, these limbs control the life-sustaining processes linking all the vital organs: brain, testes, ovary, gut, adrenal gland, heart, kidney, smooth muscle, and sensory neurons. The intriguing and unique feature of all these processes is that all are switched on by generating a simple molecule, cyclic GMP, that in turn becomes its sole functional hormonal second messenger. In this operation, the membrane guanylate cyclase is the hormonal switch and cyclic GMP is its second messenger.
Is the multi-limbed membrane guanylate cyclase signaling system specific to mammals, or does it exist also in lower organisms?
Intriguingly, this signaling system exists also in the 250 million years old silk pupae and in the mulberry plant bearing plant that the pupae feed on. Thus, the pupae and the plant has a symbiotic relationship. This remarkable relationship demonstrates that on the evolutionary ladder, the multi-limbed membrane guanylate cyclase signaling system is primordial and vital for reproduction, and the kingdoms of plants, insects, and animals are bonded.
How did you establish this astounding fact?
This was during the years 1968 to 1969. In collaboration with Dr. E. Caspi's group at the Worcester Foundation for Experimental Biology, Shrewsbury, Massachusetts, and later independently, I demonstrated that the leaves of the tea plant Camellia sinensis and Digitus lanata and the isolated fascicular cells of the rat adrenal cortex generate the common reproductive hormone named progesterone. Notably, in both cases, this hormone is vital for reproduction. I did this by first developing multiple technical tools of doubly labeled tritium C14 malonic acid, thin layer chromatography, co-crystallization, and direct isolation of the plant products. I had perfected these tasks earlier used for the isolation of plant hormones, cardenolites. Notably, I performed these isotopic labeling experiments on the tea plants grown on the balcony of our apartment in Englewood Cliffs, New Jersey during my tenure at the Thomas J. Lipton Incorporated. I did so because I did not want this activity to conflict with the official. The results were analyzed and tabulated in my friend's laboratory Dr. Bronson Aluwalia at the National Institutes of Health in Bethesda, Maryland. Therefrom, the following evolutionary related intriguing conclusions were arrived at. 1. The 250 million years old insect, plant, and animal kingdoms are evolutionarily bonded through progesterone, the reproductive hormone. The bondage demonstrated that the life-sustaining vascular processes of mammals are almost identical to the plants. The sap of the plant, containing xylem and flowing cells, substitutes blood. The former drives nutrients upward from the roots to the leaves, and the latter in the reverse order. This bondage also extends to a socially related explosive effect, birth control pill, negating the existing belief that the progesterone is biosynthesized according to the cyclic AMP signaling pathway. It was proven that the real pathway was a multi-limbed membrane guanylate cyclase signaling. To understand this topic, we enter the field of hormones.
Oh, it's a new term. Could you please explain what a hormone is?
The hormone is a regulatory agent. It is generated in an organism's cell. It is then transported via the tissue fluid, like plant sap or mammalian blood, to a distant target cell. There it manifests its physiological activity. The coinage of this term was introduced by the English physician E.H. Starling. He, in collaboration with another physiologist, W.M. Bayliss, in 1902 discovered secretin, the first hormone. Generated in the small intestine, secretin modulates gastric acid secretion and pH levels in the duodenum. Thereby, a new field of research endocrinology evolved in cellular communication organisms. Here, chemical messengers played a vital role, and the field of hormone biology started to be decoded in the molecular terms. The foundation principles of the hormonal signal transduction concept were constructed with the discovery of cyclic AMP in 1958. The core theme was the hormone did not directly target its signaling site. Instead, it did so through its second messenger cyclic AMP. For constructing the building blocks of the cyclic AMP second messenger concept, Sutherland in 1971 and Rodman and Gilman in 1994 were awarded Nobel Prizes in physiology and medicine. In this second messenger concept, hormone was the first messenger, and its interacting cell surface receptor product cyclic AMP the second. With this profound mechanistic theme, it was established that glycogen, catecholamines, and other polypeptide hormones manifested their biological activities within the cell in the same manner. Significantly, the principal features of this concept were envisioned to be applicable to all hormonal systems. The receptor was conceptualized as the critical component of a cell. There, the hormone interacted to produce a stimulus. The term stimulus was equivalent to the present term signal, which, according to the second messenger concept, meant that adenylate cyclase was the means to convert the extracellular ligand binding signal into the production of the intracellular cyclic AMP.
What the term signal transduction means?
This term was introduced in the 1970s by the Rodwell group. It meant transformation of the hormonal signal into the generation of cyclic AMP. The critical molecule involved in transduction was GTP. It bound to its signaling component termed G protein that stimulated the next signaling component, the enzyme adenylate cyclase that catalyzed the production of cyclic AMP. The G proteins existed in two forms: stimulatory GS and the inhibitory GI. The former later inhibited the transduction process. The molecular nature of the receptors specified the effect of the hormone. The cyclic AMP signaling system was thus a three-component transduction system composed of three distinct proteins: the receptor, the G protein, and the adenylyl cyclase, the enzyme generator of cyclic AMP. Subsequently, in the years 1980s, a similar G protein modulated pathway was constructed by the groups of Yasutoma Nishizuka and Michael Barrett. It was the phosphatidylinositol signaling, abbreviated as IP3. Here, the hormonal signal mobilized the intracellular calcium pool, making it its second messenger. For these contributions, these investigators were awarded Albert Lasker Basic Medical Research Award. Importantly, these two G proteins signaling pathways had a common structural feature. The hormone receptor transducer was G protein. The receptor was a 7-transmembrane spanning protein. The hormone bound this receptor and it generated cyclic AMP or IP3 via the amplified component. The generated second messenger cyclic AMP or IP3 through a cascade of biochemical resection transduced the hormonal signal into a cell-specific physiological response.
Are the cyclic AMP and IP3 the sole universal cellular signaling systems?
No, they are totally different structurally and functionally. Yet, relegated to the shadows, multi-limbed membrane guanylate cyclase cellular signaling pathway had slowly begun to rise into prominence and becoming an independent third hormonally signaling pathway. Its ascendance was, however, marked by unsurmounted hurdles. I end this video with a lingering thought on the discovery of this life-sustaining membrane guanylate cyclase molecule. Related to humanity, the year 1945, 78 years ago, on August 6, first atomic bomb was tested in US and dropped on Hiroshima and Nagasaki, respectively, on August 6 and August 9. About 215,000 lives vanished. On March 7, 1987, the discovery of the first form of the multi-limbed membrane guanylate cyclase was published in the journal Science. It sustained universal life. The years 1969 to April 2023 narrated in my next series of videos. I will begin to briefly outline the sequential steps advancing the field of multi-limbed membrane guanylate cyclase, mammalian membrane guanylate cyclase, concentrating on their molecular structure and complexity of their biochemistry and physiology. Notably, multi-limbed membrane guanylate cyclase original discovery in the year 1987 was televised by the three Memphis, Tennessee television channels affiliated with NBC, CBS, and ABC.
Thank you, Dr. Sharma, very much for this inspiring story. I appreciate this very much, and we hope to hear more of continuation of it. Thank you.
Thank you.